Hello everyone, welcome to Brain Blitz Audios. In this episode of NEET preparation, we'll be looking at previous year questions of biology in medical entrance examinations in India. The chapter that we're dealing with is the 5th chapter in the grade 12 syllabus for CBSE. The name of the chapter is Principles of Inheritance and Variation. Let's jump ahead into our first question. Given below is a pedigree chart showing the inheritance of a certain sex-linked trait in humans. The trait traced in this chart is, and our options are, dominant X-linked, recessive X-linked, dominant Y-linked, and recessive Y-linked. So we have a sex-linked trait. So what that means is that it appears more on males who are heterozygous, and for females, in order to get this disease, they have to be homozygous. So that is the property of sex-linked traits. So let's look at our options. Recessive Y-linked is option number D. Now, in this case, it shouldn't affect any females because the trait is on the Y chromosome and that affects only males. That is present only in males. But if you can see the diagram, we have almost 3 females affected with that sex-linked trait. So that means option D is incorrect. It is appearing in females as well. What about option C? Dominant Y-linked. Now in dominant Y-linked, the trait should affect all males. But over here, you can find unaffected males and affected females. So option C is also incorrect because all of the males are not affected. Some of the affected are actually females. What about option A? Dominant X-linked. Now for a trait to be dominant X-linked, it should affect everyone, because everyone who has an X chromosome, and that includes the males and the females, everybody should be affected. But you can find that there are people who are not affected from both sexes. So therefore, this trait is not dominant X-linked. So option A is incorrect. The right answer is option B, recessive X-linked. It affects females when it's homozygous, but for males it affects most of the time because they are heterozygous. Okay, so that is our first question. Moving on, the Cri-du-Chat syndrome is caused by change in chromosome structure involving deletion, duplication, inversion, or translocation. So let's look at what our question is saying. Cri-du-Cat is cat cry. So this is known as cat cry syndrome, and it affects chromosome number 5, and there is the loss of short arm. So whatever the change is present in the syndrome will have the loss factor calculated in it. Let's look at each of our options. Option D, translocation. So here the gene is present on different area of the structure. It is not lost, so option D is incorrect. Option C, inversion, is rotating the gene by 180 degrees and then placed at the right position. So there is no loss. Option C is also incorrect. What about option B, duplication? One gene or chromosome appears twice or more. So it's basically the total opposite of what the Cri-du-Cat syndrome has in chromosome number 5. So option B is incorrect. The right answer is option A, deletion. Deletion involves the loss of genetic material, whether it be a whole chromosome or a single gene. So the short arm in chromosome number 5 is lost due to deletion of genetic material. Next question. Given below is a highly simplified representation of the human sex chromosomes from a karyotype. The genes A and B could be of colorblindness and body height, attached earlobe and rhesus blood group, hemophilia and red-green colorblindness, phenylketonuria, and hemophilia. So, to verify which of these is the right option, we must know that any gene which is present on the human sex chromosome is a sex-linked trait or disease. So among our options, we need to find the option which has both of the genes relating to a sex-linked trait or disease. So let's look at each of our options. Option A, colorblindness and body height. Body height is a general trait. It affects both males and females. So option A is incorrect. Option B, attached earlobe and rhesus blood group. Rhesus blood group is due to the Rh factor, and that factor is present in chromosome number 1. And the sex chromosomes are chromosome number 23. So option B is incorrect. What about option D, phenylketonuria and hemophilia? Among these, phenylketonuria is a defect in the PAH gene of chromosome number 12. So option D is also incorrect. The right answer is option C, hemophilia and red-green colorblindness. Both of these are sex-linked diseases, and they have the characteristic recessive X gene. So both A and B can be of hemophilia and red-green colorblindness because both of these are sex-linked trait or disease. Our next question. In India, we find mangoes with different flavors, colors, fiber content, sugar content, and even shelf life. The large variation is on account of A, species diversity, B, induced mutations, C, genetic diversity, D, hybridization. Option A, species diversity, is outright incorrect because all the mangoes are essentially the same species, Mangifera indica. We might have variations in subspecies, but it's still the same species. We're not comparing mangoes and bananas, we're comparing two types of mangoes. So they are the same species. Option B, induced mutations. Mutations can cause serious chain. Mutations can cause serious difference in species. So option B is incorrect. What about option D? Hybridization. Hybridization is artificial interbreeding. And we had mangoes with different flavors way before we started hybridization. So option D is incorrect. The right answer is option C, genetic diversity. As the mango spread all over the world in different areas, it had different advantages for survival. So So they had different genetic traits because of their environment. So genetic diversity encompasses all of that. So option C is the right option. Next question. Which of the following pairs of features is a good example of polygenic inheritance? Human height and skin color, ABO blood group in humans, and flower color of Mirabilis jalapa, hair pigment of mouse and tongue rolling in humans, human eye color, and sickle cell anemia. Let's look at each of our options. Option D has human eye color and sickle cell anemia. Sickle cell anemia is an autosomal disease and it has a single point mutation. So basically, one single gene is affected by sickle cell anemia. And human eye color is also a single gene trait. Option D is incorrect. What about option C? Hair pigment of mouse and tongue rolling in humans. So this is proved to be incorrect because hair pigment of mouse is a form of epistasis and not polygenic inheritance. Epistasis is when one of the dominant or recessive characters tries to have control over the other allele of the characters, other allele of the same character. So option C is incorrect. Option B, ABO blood group in humans and flower color of Mirabilis jalapa. ABO blood group, yes, it is a good example of polygenic inheritance. But let's look at the next option. Flower color of Mirabilis jalapa. This is the snapdragon, and the snapdragon is touted as an example for incomplete dominance of one gene or allele. So it is for incomplete dominance and not for polygenic inheritance. Option B is also incorrect. The right answer is option A, human height and skin color. Human height has 3 or 4 various types of alleles, so does skin color. So both of these are good examples of polygenic inheritance. Some of the others had good examples of polygenic inheritance, but the question asks for both parts of the pair to be an example of polygenic inheritance. Let's look at this question. A gene which suppresses other genes' activity but does not lie on the same locus is called as epistatic, supplementary, hypostatic, or codominant. Let's look at each of our options. Option D, codominant, means that both alleles of a gene are expressed. An example of that is ABO blood group. So option D is incorrect. Option C, hypostatic. The question asks for the gene which suppresses another gene's activity, but hypostatic gene is the gene which is suppressed by another gene not on the same locus. So what it means is that the gene which is suppressed due to the gene given in the question is hypostatic. So hypostatic is not the right answer because it is the one that is getting suppressed, not the one who suppresses the other's activity. What about option B, supplementary gene? So in supplementary gene, the dominant gene suppresses recessive epistasis to form a phenotype. This is the one that's present in hair color of mouse. So option B implies that the gene is suppressing the epistasis to form a phenotype. So it is suppressing our phenomenon in the question. So option B is also incorrect. The right answer is option A, epistatic. Epistatic gene is the gene which suppresses another gene's activity but does not lie on the same locus. This is an example of non-allelic interactions. Our next question. X-chromosomal abnormality in human beings causes A. Turner syndrome, B. Down syndrome, C. Klinefelter syndrome, D. None of the above. Let's look at option C, Klinefelter syndrome. Klinefelter syndrome has the condition XXY in males. So it's basically an extra chromosome instead of a loss of chromosome given by XO. Option C is incorrect. Option B, Down syndrome. This again is a trisomy, that is extra chromosome of chromosome 21. Extra. So option B is incorrect. The right answer is option A, Turner syndrome, because they don't have one X chromosome, and this affects females. Now, since Turner syndrome is correct, we can safely conclude that option D, none of the above, is also wrong. So the XO chromosomal abnormality, which in humans results in the loss of an X chromosome, causes Turner syndrome, and since from 2 there is 1 missing, the abnormality affects females. Next question. A normal woman whose father was colorblind is married to a normal man. The sons would be 75% colorblind, 50% colorblind, all normal, all colorblind. So colorblindness, especially red-green colorblindness, is a recessive X-linked disease. So what it means that in a woman who is heterozygous, it is not expressed. So the woman is normal, but she is a carrier. And when she marries a normal man, the offsprings are one of the following: X-X, X-Y, XX, and XY. So X-X is a carrier daughter. X-Y is a colorblind son. XX is normal daughter. And XY is normal son. So now we have 2 sons, XY and X-Y. So what it means is that there are 2 variations for the genotype of the sons. So 50% will be normal, while 50% will be colorblind. So the sons would be 50% colorblind. Option B is the right answer, because 1 out of the 2 cases is colorblind. So that is 50%. Next question. Mating of an organism to a double recessive in order to determine whether it's homozygous or heterozygous for a character under consideration is known as reciprocal cross, test cross, dihybrid cross, or back cross. Let's look at each of our crosses. A reciprocal cross is a cross reversing the roles of parents to confirm the results of earlier cross. So option A is incorrect. What about option C, dihybrid cross? Again, this refers to crossing two lines differing in two traits. So option C is incorrect. What about option D, back cross? The back cross is a hybrid crossed with one of the parents. So it means one of the parents. So it can either be heterozygous or it can be homozygous dominant or homozygous recessive. So this can be any one of the three. So option D is also incorrect. The right answer is option B, test cross. Test cross is the name of the specific cross where an organism is mated to a double recessive in order to determine whether it's homozygous or heterozygous for a character under consideration. Next question. Bird females have the chromosome arrangement as A, XY, B, XO, C, WZ, D, WW. So birds have heterozygous females. So with that, let's look at our options. Option D, WW. This is not present. The only one that's present is ZZ, which denotes the males. Let's look at option A, XY. XY is found in human males, not bird females. So option A is incorrect. What about option B, XO? XO is found in people affected with Turner syndrome. And besides, this one is not heterozygous. This is when one of the X chromosomes is lost or Y chromosomes is lost. So option B is incorrect. The right answer is option C, WZ. As you can see, W and Z are different chromosomes. So bird females, which are heterozygous, have the chromosome arrangement WZ. Next question. Gene pool is referred to as one of the four options. We need to find out which is which. Option D, induce cell division. Cell division is induced by hormones. So gene pool does not refers to the induction of cell division. So option D is incorrect. Option C, deletion of non-essential genes. That is often found in the phrase robustness of genes and not gene pool. Option C is incorrect. What about option A, the genetic drift caused in a population? That is referred to as genetic drift itself. So option A is incorrect. The right answer is option B, aggregate of all the genes and their alleles in a population. So gene pool means a pool that contains all of the genes. So an aggregate of all the genes and their alleles in a population. Next question. Mother and father both have blood group A. they have two children, one with blood group O and second one with blood group A. So the children have one of the four types of parents. So in order to start and to understand how to go ahead with this question, we need to look at how the children are. So one of the children is having a blood group O. So O refers to the genotype IO. And the child with a blood group A can have IA, IA, and when we trace both of these back to their parents, we find that both the mother and the father had IOIA as their genotype, because only when mother and father are heterozygous do we find this scenario. So either both of them have IAIA or IOIO, or they can have IAIO, which again leads to blood group A. So either way, we can find out that both parents are heterozygotic and they have IA and small I. Note that small I is the same as IO, and that refers to O. So option D is the right option. Now, our next question. When one gene controls two or more different characters simultaneously, the phenomenon is called as apomixis, pleiotropy, polyploidy, polyteny. Polyteny, option D, is referring to DNA replicated without separation. So in anaphase, due to some error, the DNA is not separated. So option D is incorrect because polyteny is not the case where one gene controls two or more different characters. What about polyploidy? Polyploidy is where the individual has more than two sets of chromosomes. So option C is incorrect. What about option A, apomixis? So this is asexual reproduction resembling sexual reproduction. In this case, no fusion takes place. So again, apomixis is the incorrect option. The right answer is option B, pleiotropy. Because pleiotropy is where one gene controls two or more different characters. Let's move on to this question. Three children in a family have blood types O, AB, and B respectively. What are the genotypes of their parents? So let's represent small I as the one for O, IA is for A, and IB is for B. So the children have blood types O. So the first one has O, so it is small I, small I. The second one has AB, so it will be IAIB because both of these are codominant. And for the second person to have to have both like this. So we just need to find out O and AB in order to understand that both the father and the mother need to have at least one of both. So basically what this diagram means is that the parent either has IAI or IBI. And so that makes sense because if we have a scenario like that, then it's easy to pair up these two in order to form an IBI genotype. And that refers to the B blood group in the third child. So for the parents to have all three of their children in the same order, we need to have the genotype as IAI and IBI respectively. In the second option, only A and B are present. In the third option, only AB is present because both are homozygous. And in option D, only AB and A are present. All of these are incorrect. Next question. If both parents are carriers for thalassemia, which is an autosomal recessive disorder, what are the chances of pregnancy resulting in an affected child? So let A be normal autosome, and let small a be thalassemic autosome. So we know that thalassemic autosome is recessive from the question, so normal will be dominant. So the question says that both parents are carriers for thalassemia. And so since thalassemia is recessive, it is just required to have one dominant allele beside it in order to negate the effects of thalassemia. And since they said carriers, so this is their genotype, capital A, small a. On crossing these two, we could use a Punnett square and we can find out the genotype of the kids. So again, we have dominant, recessive, dominant, recessive. So these two come together to form this. These two come together to form capital A, small a. These two come together to form capital A, small a, and the two thalassemic autosomes come together to form small a, small a. So in this situation, we find that capital A, which is normal, is 1/4 of the total. And capital A, small a, which is carrier, will be 1/2 of the total population. While small a, which is the affected child, has a probability of 1/4. So that means there is a 25% chance of a pregnancy resulting in an affected child, because 1/4 times 100% would give you 25. So option B is the right answer. Next question. In Huntington's disease, the unaffected persons are homozygous for normal allele small h. The following diagram here is erroneous because of one of the 4 options. We need to find out which of these is the right option. So the Huntington's disease have the characteristic. So Huntington's disease have the characteristic of being autosomal dominant. So that means for the gene containing Huntington's disease, it is dominant. So when it is dominant, it is affected and the unaffected have the small h, which means it is recessive. So in this diagram, we find that this person number 1 is unaffected. So she has small h, and the person who is affected has capital H, or capital H, small h. But according to our question, it is given that they are homozygous parents. So let's go with the assumption. So we find that all 3 of the kids So we find that some of the kids have Huntington's disease while the others do not. But if you look at the right section, where people 6 and 7 give birth to 10 and 11, we find that person number 11 has Huntington's disease. So in order for this to happen, at least the genotype should be HH. And this should happen when either 6 or 7 should be affected. And so we can find out that either person 6 or 7 should have the disease for individual 11 to show Huntington's disease because it's autosomal dominant. So that is the right option when you analyze this diagram. Now, option A means that it shows both male and females are affected by Huntington's disease. That is due to autosomal dominant, which is correct. So this statement is true. So there is no error because of that. At least 2 of the children, 8 or 9, should have the disease is option C. So when it is the case of heterozygous parents, we can have the case where both of the children can have small h. So in that case, they won't be affected. So option C is also incorrect. And so since A and C are incorrect, we can safely conclude that D is also incorrect. The last question. The experiment shown in the given figure has been carried out by Morgan to show the phenomenon of linkage and recombination. In cross 1, genes are tightly linked, and in cross 2, genes are loosely linked. Then what will be the percentage of recombinants produced in cross 1 and cross 2 respectively? This is the diagram for the cross, and our options are the following. So before we go anywhere, we should know that when a gene is tightly linked, it means that there is less percentage of recombination, because the percentage of recombination is directly proportional to the distance. It actually refers to the distance. So when a gene is loosely linked, there are more chances, so there is more percentage of recombination. So this is the first point. The second point is that linked genes are always having a frequency which is less than 50% of recombinant frequency. So the recombinant frequency of any linked gene, whether tightly or loosely, will be less than 50%. So with both of these in mind, let's go attack our options. Option A, 98.7% and 62.8%. these are more than 50%, and which means that both the percentages are for parental genes. So option A is incorrect. Parental DNA is 98.7% and 62.8%. What about option D? It again has the same problem. It has both the frequencies more than 50%. And when it's more than 50%, it is not recombinant frequency. It will be parental. What about option C? 37.2% and 1.3%. Both of these are less than 50%, but there is one hitch. Remember our first rule. Tightly linked means less percentage of recombination. Loosely linked means more percentage of recombination. And over here in cross 1, they are tightly linked. So that means cross 1 should have less percentage of recombination than cross 2. But in option C, it's 37.2 is greater than 1.3. So option C is incorrect. The right answer is option B, 1.3% for cross 1 and 37.2% recombinant frequency for cross 2. So that ends this episode of NEET preparation. We hope that you found this video interesting. For more of our useful content, please visit our YouTube channel, Brain Blitz Audios, and subscribe to our channel. We'll be back with more interesting and useful content such as learner words, mind maps, NEET preparation, CBSE board exams, etc. So until then, take care, stay safe, bye-bye for now.